All right. Yeah, let's start my presentation today. Um, it's a great pleasure to present my previous paper, Nature Neuroscience 2019. The title was the profiling allele-specific gene expression in brain from individual with autism spectrum disorder reveal preferential minor allele usage. Let's start with the introduction. So what is the autism spectrum disorder? So this ASD is a concrete, including so many different ASD like the disease pattern. So that's why we call it the spectrum disorder. Also, ASD has a common feature. It's a communication problem and repetitive movement issue, as well as a social interaction problem. And the prevalence of ASD is pretty high, so about 1.5% in poor population, so quite high number. Also, the most common ASD is a D15q, it's about 3% of ASD patients has a duplication of unimprinted region. So that's because this is strongly linked with the imprinted region. I was interested in the study of allele-specific expression ASE study. So what is ASE? So this is a human chromosome. So if we look at the one chromosome here, and the one strand is from the dad side, the another one is from mom side. And then we can see homozygote pattern as well as heterozygote pattern. So heterozygote pattern can have some deletion mutation like this G allele. So it sometimes can be very dangerous for the human health. So when we check the expression, the homozygous allele only express only A allele, but heterozygous allele also gonna have a two different allele expression. It's a G and A, but their ratio is not always 1-to-1 ratio. Sometimes 1-to-3, sometimes only one is expressed, the other is not. So here, G allele expression is kind of restricted, right? So ASE can buffer some deletion mutation like a G allele. So it's not always true, but it's a kind of possible mechanism behind the ASE. So I kind of focused on kind of this kind of a buffering the kind of mechanism of ASE in my talk. So for this study, I used the post-mortem human brain sample. So my target region is the BA9, it's a prefrontal cortex of human brain. And then BA 41, 42, 22, kind of around this area is the temporal cortex. As well as I consider the Burmese is the cerebellum area. So from the brain sample, I generated analytic data. And this is the number of the sample that I got generated. And then cerebral data and the small analytic data. So using this multi-omics data set, I generated my data process. So first step is I figure out where is ASE, right? But as she mentioned, Maria mentioned before, this is a very challenging, the kind of a pipeline is behind to identify the ASE. So first thing, also the very important thing is the reference bias. So this is the reference sequence. So this A earlier is here, but this is the kind of point of the SNP. So A and T, the SNP. So if the analytic read has A earlier here, so they're gonna map very well without any issue. But if the site has a T earlier, so that time these two gonna have a fail to map. So it's gonna have some disadvantage. So if I can mask to the third earlier, like a G, it's not belong to any of them. The case, both of them gonna have the same disadvantage. I can expect that they might gonna have a 1-to-1 ratio instead of the biased, right? So it's the kind of reference bias was already studied. They found that this group found about 40% of a false positive signal of is from this reference bias. Actually, there are top ASC signal. So I address this issue for my pipeline. So this is my pipeline. So 263 analytic data as prepared. And then I also prepared the masked reference. So here I collect all of possible the SNP information from so many different sources. So I targeted about 40 million SNPs and then map based on the masked reference, I kind of mapped all of the read, and then I filtered out reference bias, the SNP. So also kind of artificially generated a test set, and then I evaluated all of the SNP location, and then I filtered about 10% of the SNP. And I did the genotyping for all of sample for the 40 million SNPs, and also I checked the ancestry of each sample. And then I did a group-based ASE study as well as individual-based ASE study. Group-based ASE study means kind of combine some sample based on the group, like a control and the tissue level, and then the ASD patient, and then combine all of them and then figure out how they look like. In that case, I consider the linear mixed model because they have so many different factors that we need to consider. And then after the test, I did a multiple test correction after considering LD-based SNP pruning, because of some crossing across the SNP has a kind of very similar kind of feature, right? So that's why I need some pruning. Also, finally, I filtered out indel-inducing reference bias. So here I consider only sequence-based, but there are some or more the reference bias based on indel. So I filtered out as well. Also, for individual-based ASE study, I use the Fisher eject test, and then I follow the same thing as well. So first, I considered what's a common ASE pattern in brain tissue. So this is ASE allele BA9, BA41, and Burmese. So they are very highly overlapped in general. So I believe that they are very conserved. Okay, and but when I did the gene enrichment study with them, they have very highly enriched the ASD risk gene. As you see here, imprinted gene also highly enriched, and then FRP target, HUR target, RD-FOX1 target, so also highly enriched. So they are well known, the ASD risk gene. Also, when I consider the rare de novo variation data set, so also all of them showed high enrichment with the ASD. So since this ASE is showed high enrichment with the ASD risk gene, I believe the ASE can mitigate deleterious mutation, so kind of a buffering, the kind of a dangerous kind of mutation effect. So I moved to the kind of a comparison. So I studied the different AS pattern between ASD and control here. So the control, the ASE allele, and ASD, ASC allele. So as you see here, the ASD shows a kind of more tissue-specific pattern. See, the less, very small overlap compared to this, right? 10 times less, right? 12 times overlap. And when I compare SD-specific ASE, the Burmese show kind of a slightly different outlier compared to the other cortex sample. So that's why from now on, I mostly focus on cortex sample as a combining BA9 and BA41. So this is from now on, it's only cortex data, data process. This is all of ASE kind of result. So I show the p-value and generate the Manhattan plot. So you can see here, from cross genome, we can find the ASE, the pattern kind of earlier, all over everywhere, right? But when I compare their kind of ASD between control and ASD. ASD shows a kind of small number. So ASD kind of the pattern is kind of decrease compared to control. So this pattern also showed when I studied individual-based ASD study. So the red is SD and blue is the control. When I consider SE rate, right? So meaning the high rate means more SE compared to the others. So SD patient has showed a very small SE rate compared to the control. So it's also a double compound. So general conclusion is SE is kind of less in SD. So SD has a more risk for the mutation. So if the SE is kind of beneficial to prevent the deleterious mutation, okay? But those studies had some limitation. So when I compared between SD and control, we need to consider only common allele, not rare allele, because and it's a two samples should have the same SNP, right? So I want to compare the what's the common allele kind of things. So rare allele things. So that's why I consider the quantitative allele expression study. So I calculate the minor allele expression fraction. Okay. So and I generated this kind of a density plot for control and SD. And then when the minor allele expression increase, and then shows a kind of this high expression here, and then no expression here. So based on the density plot, this is bottom is a balanced expression, and then these tails are imbalanced. And also very end shows a monoallelic expression means only one allele expressed, not the others. What interesting is there's a control shows a more high expression here, but in imbalance is a high in imbalanced portion, the S is a more high expression, and then this tail as an shows kind of unique pattern. So here also monoallelic expression, the high expression shows high expression here, but here is the opposite. So kind of in ASD, kind of the ASD, ASDE, allele-specific expression is kind of unstable. So that's why kind of spread out. Especially the minor expression is a kind of strongly showed up. So in general, when the minor allele frequency is going down, the preferential, the major allele expression is rise up. Okay, and then the preferential minor is going down. So it's also showing kind of the opening kind of a pattern. So when I compare the major and minor allele expression between the control and the SD, SD shows slightly more minor expression. So statistically, I can tell SD has a proportional minor allele expression, but it's not very strong. But when I consider the monoallelic expression, these two tail, especially at it's a very highly enriched at rare SNP. The time this is the monoallelic expression, it shows a very high monoallelic expression in SD. So this is kind of a big problem in SD because the minor allele monoallelic expression is kind of very deleterious in general. So it's not predictable or expected one. So that's why it might can cause some disease effect in SD. So from now on, I studied the monoallelic study in SD and 252. So here, I showed the SNP location in coding gene in control sample. So interesting one is major monoallelic expression shows a very high possible amino acid changing and then a high possible loss of function mutation compared to minor monoallelic expression. It means in control, there are the kind of risk mutation is highly kind of mitigated by SE. Also, when I did the enrichment study with the monoallelic expression gene, in control, the major allele monoallelic expression show high enrichment with the sulfur gene as well as other as the risk gene. The sulfur gene is a very well-known, sulfur gene is a well-known risk gene. So many patients kind of share the mutation. And but in SD patient, this enrichment is kind of shifted to minor, meaning the problematic, the risk SD risk gene can have some mutation. So the kind of a mutation like the expression is not mitigated at all. Also, this pattern also show in duplication patient as well, duplicate Q. So it's a kind of a very kind of a dangerous for ASD patient. So when we look at the distribution of the monoallelic expression across the genome, so it's a minor allele monoallelic expression here and the major allele monoallelic expression bottom. So minor, so kind of it's across the kind of all over genome, but when we make kind of this kind of a figure, so there, the minor allele monoallelic expression ratios are pretty high at chromosome 14 and 15. It's kind of interesting. Not the other place. Only these two places extremely high compared to the others. So I kind of what problems happen whenever the major allele changes to minor. So I look at the allele shifting pattern here. So one possible mechanism is a minor allele can change to major, also major allele can change to minor. So here is shows the kind of a location. So only few spots here. Now both of them shows previously reported in SD. Here's all over SD. The patient that can have some issue with the deletion and the duplication. So those areas are quite important for in SD. And when we consider kind of common area, so mostly highly reached in early shifting region is only pointing two region, 14q32 and the 50q11. The very small region is a 0.3 mega and then 0.2 megabase. And then they have so many kind of early shifting. But one unique feature of these regions is they are both imprinted region. Okay, so it's a maternally expressed genes, so all paternally expressed gene. And another interesting thing is all of non-orphan CD box SNO RNA exist only in these two regions, not the other genome at all. Only two regions has all of orphan CD box SNO RNA. And then it also has a very interesting feature. It's a very brain specific expression. Also, it also induces alternative splicing of its target gene. It's a very unique feature of compared to other RNA. So what's the splicing changes means? So when I consider RNA target splicing change here, so the target is the most highly enriched splicing change in ASD is higher than non-target. See the ratio is higher than no splicing target. Also, the same thing happened in DU50Q patient as well. It's pretty high compared to here. So when I do gene enrichment study with the SNO RNA target gene, splicing change in target in ASD has high enrichment with the sulfur gene, yeah, as well as other ASD risk gene, and but very weak enrichment in no splicing target gene in ASD. The same pattern that also showed in DU50Q the patient. Also, when I consider rare de novo variant, only these two shows a high enrichment with the ASD patient, ASD enrichment. So the possible minor early expression, possible minor early reference preference effect in ASD is showing kind of this way. So if some deletion mutation happen, so G is a minor allele. So ASD can buffer this deleterious mutation. So if it's a successful, so the people gonna be healthy and the sooner they're gonna have a normal splicing. But if it fail to buffer it, sooner they're gonna do kind of abnormal splicing. Also, due to the mutation effect, it also can contribute the SD as the kind of feature. So here's my conclusion. So SD exhibit reduce the SD and then show more tissue specificity in SD. Generally, major alleles are preferentially expressed. The preferential major allele expression is a mechanism for buffering deletion mutation in protein coding genes. However, SD has a more minor allele expression than control, so it's indicating a potential disease mechanism involving the reduced minor allele buffering. Also, minor allele monoallele expression gene in SDs are enriched with the SD risk gene. Also, common allele-specific rich region in monoallelic gene, monoallelic expression at 14q32 and 15q11 contain all of the orphan CD boxes RNA that kind of change the alternative splicing. In SD, the RNA target gene shows a more special change. And then their special change genes are strongly enriched in SD risk gene and contributed to SD pathogenesis development. This all of my work is done in UCLA under the Daniel Gashin's kind of advisor, and then I'm currently in UT Southwestern at his lab, and then this is my research support, and then this is award during kind of this project. Thank you very much. That's all what I have now.
Anyone have a question?
Yeah, thank you, Chang Hoon. That's a very interesting talk. I'm wondering what do you think is the reason that um, that to begin with that the autism cases are showing this different kind of ASE patterns overall? What's the most upstream change that led to this?
That's a very good question. This is not very unclear. So as the Maria mentioned, also the promoter, base, the kind of controls kind of behind it. Also, there are so many different things. As you mentioned before, the RNA decay is one of the things. So also some methylation, gene methylation is also can control it. But those things people are still kind of studying now, but very challenging project.
Yeah, because I'm just thinking about the circularity here a little bit because sometimes you think of autism partially can be caused by de novo mutation, and supposedly that's the original cause, but then it seems like almost like the ASE is more upstream of at least some of the de novo mutations, or
Yeah,
maybe there's some feedback loop there, right? But
yeah. I believe for the paper, I believe the ACE is a mechanism detects some mutation, and then they just kind of a beneficial effect. So you're right. There are some that kind of a feedback loop behind,
I see.
but no one studied much. So it's gonna be my future study.
Chang, I'm curious about the snow RNA
Mhm.
locus. So for autism risk, what is the variant there that is causing risk for autism?
Also, can you repeat your question again? I kind of
The I don't know if I understood well the your story about the snow RNA. So the snow RNA isn't a risk locus for autism. Is that correct?
yeah, snow is very enriched in early shifting region. So in where the in genome there it has two spot has a very high SDE, the fluctuation, the kind of it used to be the expressed the reference earlier, but it's in SD patient, an alternative allele only expressed. Those kind of weird hotspots exist in human genome, only a two spot, and then in those spot only small RNA exist because of kind of this early specific expression change. Their expression is kind of very fluctuated. So I didn't show this time, but it's a super fluctuated. And then they are kind of alternative expressing the mechanism and kind of disrupted. So I believe that's why it caused ASD. I wonder if it can be a good answer for you.
Okay. So, yeah, thank you. And what are you planning to do more things with this data set? There, I guess it's a rich data set.
Actually, once I moved to UT Southwestern, I'm currently working on some sleep study now. So it's still SD field, but not exactly allele-specific expression.
Okay.
I'm also curious about what do you think about the variability across the brain region, especially in autism compared to the control cases?
So it's a very interesting and good question. So it's not clear because I just found the SD has a more variation, the tissue-specific pattern. I believe because SCE kind of the function that is kind of a buffering kind of deletion mutation is kind of disrupted. That's why they got their kind of a common feature of SE pattern is the kind of different across the brain tissues. Is how I can guess, but nothing very clear. So abnormal SE pattern. That's kind of randomly jumped out. That's why the tissue specificity is a kind of an increase.
Right, probably less control. Yeah.
Exactly.